throw a thumbs up, please. Cool.
So I think I'll put some light.
Please do. Good to go? All right, guys, I think we're live. So thanks everyone for joining. I'm joined here today by Rashid and Dimitri, the researchers behind the first project that's being listed on Catalyst, as well as Michele, who's representing the VitaDAO side. We're really excited to co-host this event alongside the VitaDAO team. So as you know, this week, Molecule launched Catalyst, a platform that democratizes research funding, allowing individuals to directly support projects that resonate with them. This decentralized model aims to rectify a lot of the inefficiencies with current research funding and that entire apparatus, ensuring that funding better aligns with societal needs. The objective of Catalyst is to empower individuals to use their crypto to make impactful funding decisions, fostering a more transparent and impactful research funding system. And then I'll hand it over to Michele to say a few things from the VitaDAO side.
Yeah, absolutely. Thank you, Benji. On behalf of VitaDAO, I want to say that we are very excited to join forces for this session. As you know, I personally source this project, and I'm very excited about it because I think it focuses on an area of aging research, translational fidelity, that has been under the radar for a while and now is attracting again a lot of interest after the paper from Gorbunova and Gladyshev, I think in 2018. And it aligns with VitaDAO's goal of funding moonshots that are projects that are high risk, high reward.
Yep. So anyway, broadly, our goal with these sessions, these AMA researcher sessions, is to create an open forum where researchers can share to the broader DeSci community their project, go a little bit deeper into the science, and get pressing community questions answered. But before jumping in, I did want to remind everyone, not everything on this call should be, nothing on this call should be considered investment advice. We don't endorse any of the claims made by the researchers. Their opinions are their own, and we provide this platform solely for discussion purposes. So with that, we have the privilege of welcoming the esteemed researchers behind Project Transfidelity. So yeah, Dimitri and Rashid, please take a moment to introduce yourselves, maybe explain how you guys met and how you came to this line of research.
Okay, Dmitry, maybe you go first.
Well, okay, so I'm almost 50 years old. Now I studied molecular biology and biochemistry at the Moscow State University in the former Soviet Union. I got my PhD in the Moscow State University in 2000. And since then, I worked in Europe as a molecular biologist, first in Austria, and then starting from 2007, I'm permanently working at the University of Zurich in Switzerland. Mostly it was Institute of Medical Microbiology, where I focused on development of new antibiotics, so antibacterial drugs, and also studying mechanisms of resistance of those. And then for some reasons, which we probably go through further, we moved from these antibacterial therapies toward the aging and anti-aging and age-related diseases drug development. That's more or less all about myself.
Okay, so I'm originally from Kazakhstan, studied at the beginning, I think for 3 years, I studied in Novosibirsk University. It also was former Soviet Union. Then I graduated from Almaty University, State University, and then I started to work in Almaty, also this former capital of Kazakhstan, for several years. I worked in the Institute of Molecular Biology, and then I did PhD there, with partially there, some partially in Europe. Finally, at the beginning of 2000s, I finished my PhD and then went for postdoc to Switzerland and to mostly work there. Also, I worked shortly in some other countries, but mostly I stayed last, let's say, 25 years in Switzerland, working in different institutions. And now I'm working in Biocentrum, it's Basel University, as a senior researcher. And mainly I was working on translation as academic scientist. And it was I started as a plant science researcher, then I went to bacterial research. Finally, we met with Dmitry in the Zurich University and worked there for 15 years in the laboratory of Eric Böttger. It was a very nice time. And there we finally came to this project. Yeah, and then maybe I will later tell how it developed further. Yeah.
Great.
Go for it,
Thank you
Michele.
so much, Rashid and Dimitri, for this overview of your background, which is extremely impressive. And I just wanted to give a brief overview of let's say, of the topic that you're touching upon, and that's improving your project focuses on improving the translational fidelity of protein synthesis. If I correct me if I'm wrong. And I wanted to and how this is also related to the development of Alzheimer's disease. I wanted to ask you, what's the story behind the research and what led you down this research topic?
Okay, maybe I start. So as I told, and Dmitry told as well, so we mostly focused on protein production in living organisms, yeah, in translation. So Dmitry really deeply studied structure of prokaryotic ribosome. I worked in plant ribosome. And then I also worked in some plant virology and studied why, for example, virus invading cell can outcompete other mRNAs, become translated and occupy complete translational machinery in plant cell. And we were moving in parallel. And then we met as I said, in Eric's lab in Zurich University. And there his lab was focused on drug design, on development of different antibiotics. And one of these antibiotics was aminoglycosides. This is very interesting, very cheap, very efficient antibiotic. It's second line defense against different infectious diseases like tuberculosis or some other diseases. And this drug has interesting effect on bacteria. You can imagine, there are some drugs which can stop growth of bacteria, and some can just kill. So it's bacteriostatic and bactericidal drugs. And then we realized that one of the reasons why aminoglycosides are bactericidal, so killing bacteria, because they, in addition to suppression of translation, they can also induce misreading. So it means when you have very high level of misreading even in bacteria suffers a lot. So from this point, we were developing different new aminoglycosides. And of course, when you develop drug, you want to reduce side effects of the drug. And in parallel, you also want to increase efficiency of the drug. So, and we realized that one of the side effects of these aminoglycosides in human body is causing ototoxicity and nephrotoxicity. Then people who have this anti-tuberculosis drugs like amikacin, this can be used against tuberculosis, they can become deaf, or they can have problem with their kidneys. And one of the reasons which we realized that people who have special mutation in mitochondrial ribosome, they have higher sensitivity to this drug. And one of the effects of this higher sensitivity is increased misreading in mitochondrial translation. So then we start to focus more on this effect when different drugs which are really toxic for human have this side effect like increased mistranslation induction. And from this time point, we start to look more on the effect of misreading in general. And this became our real topic. We gained a lot of knowledge in this. We created different models where we could induce in living, even mammalian organisms like mice, we could induce mistranslation and observe the effect of this. And we saw that the higher mistranslation is, the more aggregation you have in cell, the more let's say very, very dramatic negative effect it has on living organisms. So this is how we came to this topic.
Amazing,
Wow.
amazing.
Well, I guess that kind of gave an interesting insight, which is, I know that you guys had previously, researched into antibiotics, but it was really on the same axis of translational fidelity. You had just realized that decreasing translational fidelity killed microbes. And so the next thinking was, oh, if we can decrease or increase translational fidelity, we could potentially have a positive effect.
Absolutely. Yes.
Very interesting. Cool. Sorry, I didn't mean to interrupt. Go for it, Michaela.
Oh, no worries at all. That's actually very interesting. And can you tell us a bit more what the evidence exists in the literature for the association between translational fidelity and the various age-related diseases?
This probably I can answer. This topic is not new. Originally, this idea was first formulated in 1960s as a so-named protein catastrophe or error catastrophe hypothesis. At that time, it was suggested that if you have errors during translation, then since your ribosomes and all other components of the cells consist of proteins, so the more mistakes you do, the more mistakes is included into the ribosomes and other components of translation apparatus, and the more mistakes they do in the next round. So it's somehow autocatalytic process, and at the end, you have complete abnormality and death, actually. And later, it was not even disproved. The people just moved to other topics, to the mutational theory of aging and other stuff, because initial trials to prove these hypotheses, they were not successful, mostly because the sensitive used at that time was not sensitive enough from one side. From another side, it was not possible to measure translation fidelity in living organisms, so in real organisms. And the people tried to use senescent cells or other cellular models to prove the hypothesis. And as we know now, it's not proper models, actually. So senescent of the cells, it's a completely different mechanisms, completely different process. So it's not similar to the real aging in real organisms. Anyhow, the interest was waked up in the late 2010s. And in a recent time, it was several publications. We can proudly say that quite some of those publications came from our lab. And there are also other publications from other groups that in reality, so that the translation fidelity plays extremely important role in the aging and age-related diseases. Because so what happens with aging, actually, is you have two processes which go toward different directions. First, your proteostatic machinery, this is proteasome and autophagosomal lysosomal system, which is dealing with wrong proteins and other garbage in the cell, they goes down. So the activity just goes down with the aging. And another process which is not described yet, but now we are publishing the paper where we so clearly show this, that your fidelity of translation with aging goes down. So you have more error-prone ribosomes, which is actually more or less obvious. So everything with aging goes down. So what can get worse, getting worse. And So those two mechanisms together, they lead to the accumulation of the garbage inside the cells in the form of first of the protein aggregates. That the proteins which cannot be, so they're not functional, and when the cell cannot remove it so properly in time, then those protein aggregates start to accumulate, and then the cell has a huge problems. So that's why a lot of, I would say probably most of the age-related neurodegenerative diseases, they're all related to aggregation of specific proteins. So I must say that you should not be leaded, so you should not understand it wrong. Because when, for example, people say that, for example, alpha-synuclein plays a major role in the Parkinson's disease, it does not mean that aggregates, if you check them, they consist only of alpha-synuclein. No. There are actually all possible cellular proteins inside those. The protein aggregates, which means that it's general process. It's not specific for certain protein. It's just general cellular protein. So you have problems with protein aggregation. And major role plays here the misreading, so mistranslation, so erroneous translation. Because so normally ribosomes work with mistakes. So ribosomes, when ribosome doing new proteins, it makes mistakes. Yeah, so let's say 1 out of 3,000. 1 out of 3,000 amino acids in your proteins is wrong. It looks like not too high, but we checked it specifically. If we increase it just twice, the mice with this increased mistranslation have severe problems with health. If you increase it 3 times, it is already death. So it's embryonically lethal. It means that it's very, very important to keep this threshold actually as low as possible. And when you increase it, you have problems with health. And so we published at least 3 papers in high-ranking journals about the effect of mistranslation on aging. It's 2021 Science Advances, then 2021 also Nature.
Communications biology, I think, yeah?
Yes. And the third one, communications biology, yeah. And third one is Cell Reports. It's about connection between the decreased fidelity of translation and possible symptoms of Alzheimer's disease. There are also some other publications. The major one is so very broadly cited from Cell Reports in 2021, where the people tries opposite. So what we did, we increased error ratio, and the people in this publication, they try to decrease, so make translation more precise. And what they observed improvement in the health of model organisms. In that time, they used primitive organisms with very short lifespan. So it's fruit flies, Drosophila, and worms, C. elegans. And in both cases, they observed that those organisms have a beneficial effect. If they decrease translation error rate, all of those studies were done with specific mutations. So it's well known since a long time that you can mutate some ribosomal proteins so that the ribosomes are starting to be more precise. And yeah, so what is our idea in frame of this project? We want to try to find actually the compounds which are able to do the same, because we know for the decreased fidelity, we know that those compounds exist. The best known probably is geneticin. This is well-known aminoglycoside antibiotics, which works also on eukaryotic cells, which significantly decreases error rate, fidelity. And our idea is that also should be compounds which have opposite effect, which are able to increase fidelity of translation. And we expect from those compounds highly beneficial effects, especially we expect reducing of the mistranslated, misfolded, and aggregated proteins. That's
Amazing.
actually so more or less summary.
Thank you so much, Dimitri, for this thorough review of let's say what has been done in the past and your work on this topic. And I find very interesting, especially your take on how let's say like the theory of aging associated with with improving translational fidelity has not been actually disproved. It's just that there were issues at the time with how to measure translational error. And actually, it as you said, it's great that this topic has found again a lot of interest in the past few years, and I'm sure that a lot more and more research will come forward on this topic. And
that's for sure, yeah.
Yeah. And I wanted to ask you guys, like you have given already thorough overview of what you have done so far on and how this project is not only related to aging but also to neurodegenerative disorders. What's your goal and what's the funding? What does the funding aim to cover? Like the funding for this project aim to cover that they will be on Catalyst.
Okay, so probably I answer this. First of all, we had, let's say, during our, let's say, 15 years of work on this field, we, as Dimitri already summed it up, so telling that we realized that the misreading should have some effect on, let's say, proteostasis in the cell. And our goal was, let's say, we thought maybe we can do it like other way around. We increase fidelity, decrease cost of, let's say, this effect on cell. So, and there are actually two possible approaches for this. Yeah, either as it was described already in this paper, either you introduce mutation in ribosome, make this ribosome more precise, or you apply certain drug which does the same thing. And our idea was let's try to find such a drug. So to do this, we first of all developed screening platform. So this screening platform which consists of two units. One unit allow us, let's say, one platform allow us to do screening for in vitro translation, and second one allow us to screen compounds in cell culture system. In both cases, we can determine fidelity of translation. And using these two platforms, we did screening of libraries of different compounds, of more than 2,000 compounds library. And after screening, we identified 5 compounds, 3 compounds using in vitro screening platform, and 2 compounds using this cell culture. And this was our starting point. So we tested this unfortunately these 3 compounds which were isolated using this in vitro system were also having additional effect as inhibitor of translation, because you make more and more precise translation, you directly affect ribosome, and then finally ribosome really stops because it doesn't want to make any more mistakes. Yeah, you just forbid it with higher concentration. And then these compounds, we call these direct compounds, these compounds which directly interact with translational machinery. Yeah. Other 2 compounds which were working at the level of living cell, we call them indirect compounds because it didn't work in vitro system. So it means it does something to the cell, and then cell reacts such a way that fidelity of translation gets increased. Yeah, and by now we don't know what happens in cell because of this compound. But these 2 compounds we consider as the most promising ones because they don't induce stoppage of translation, so they don't cause a problem for translational machinery. It even helps it probably because then translations start more precise and work better. So, and these two compounds, we decided to develop further. So our next, let's say, goals. So we tested, of course, these two compounds were also tested in so-called aggregation assays. When you like have system where you in this system you have cells which in normal condition or because of different treatments they start to form aggregates of proteins, and you can register it by one of another mean. Yeah, so you can, it could be fluorimetric or it could be other like colorimetric stuff, whatever. Yeah, you just detect a formation of aggregates. And we did it in vitro with some mammalian cells, and these two compounds had positive effect. So they suppress formation of aggregates in this system. So which is our next step, which we made. So now what we want, yeah, what we need money for. So we need to, let's say, round up the so-called proof of concept. We want to show that these two compounds can work and suppress aggregation in more relevant to pathology cells, like human cells, like human neurons from sick people who had some one or another neurodegenerative disease like Parkinson or Alzheimer or any other kind of. And these cells we are planning to treat with our two promising compounds and prove that, or check at least, yeah, whether they can suppress aggregation formation. This is actually first question which we want to answer using your money. Second question would be really important one. What these two drugs do? What is the counterpart of the inside cell which interact with this compound, and how this finally translation fidelity gets increased? This actually very fundamental scientific question, but without answering this, we cannot proceed, because this question, answer to this question will allow us to make wider, let's say, variety of these compounds. Because what we started with, it was like partially relying on luck. Yeah, we screened a huge number of compounds, and we were lucky to catch two. Now we have them. Yeah, we have something which is really there. Now we need to go further. We know where we start from, and we need to find counterpart. We need to find mechanistic effect of these compounds. We find the protein which or enzyme which interacts with our compound. Then when we know the target, we know the pathway. When we know the pathway, number of targets gets increased, because we then in this pathway we can interact with any of the downstream participant of this pathway. Then we can really narrow down our effect. We can increase it, make it more efficient, more specific. So from this point, it's really identifying of the target is really vital for this project. So, and this is second thing which we need to clarify using your money. And yeah, sorry, you want to?
No, I have a question on exactly that, which is,
Yeah.
as I understand, the compounds that were screened were all FDA approved.
Yes.
So are their targets not published already, or do you believe that the effect that they're having here is off target relative to what they were originally designed for?
It could be that there is an overlap, because the drugs were used for treatment of some other diseases. So, and it's not related to neurodegeneration or aging, whatever. And it could happen that there is some kind of meeting point, very decisive receptor or whatever which interacts with this drug, and then it's normally when you induce some receptor, the downstream effects could be very various. It's like cascade of different kinases which activates a lot of different, and one of the final target became ribosome, or maybe ribosome or some translational factor, or maybe whatever tRNA synthase, whatever. We don't know this. We just need to know this. And there is very nice method to identify. It calls PISA. This is like a very well established method, which is even in my current lab, people are widely using. It's very good method, and we believe that we will be able to identify the target. So, and then just to round up, so the third thing what we want to achieve within this project is we want to make like after identifying the target, after identifying the pathway, we can now discuss this result with people who are more related to this business of drug development, who can support us with their expertise and give us let's say hints which way we should go. Yeah, this is like consulting and modeling work. It also needs some time and resources.
Yeah, absolutely, Dimitri and Rashid. It's I think that as DAOs and molecule, like we have like our main goal is to support you with these topics, with these that you as you are great scientists. Of course, you are not experts in regulatory or business development, but like that's what DAOs are here for, like to help you figuring out these unknowns and to support you with aspects that are not related, let's say, to scientific development of the projects. And so, would you like to please stress once again why the funding prioritizing pursuing the MOA before medicinal chemistry, as this was something that was brought up by some community members of VitaDAO in our governance platform. Why do you think? What did you determine it was better to discover the target over going straight to compound optimization?
So, Dmitry, you want to answer or me?
Could you please formulate it once more?
So, yeah. So basically,
the final question. Yeah.
Yeah, like what did you determine that it was better to discover first the target rather than pursuing medicinal chemistry optimization, rather than vice versa, the first medicinal chemistry optimization and then pursuing the target?
Well, in principle, we can do it in parallel, of course. But we think more important is identification the target because of IP. Since all those compounds are already so published and patented, so one of the possible way is to identify the target, and then the best way to identify the whole pathway. So, and then to go for another targets with probably another compounds which could be not IP protected. So that's one way. Another way is that I think actually it's enough already. Maybe you, Rashid, want to add something.
Yeah, I can. Why is important to identify the target? Yeah, when you have a target, you have a molecule interacting with this target. Yeah, normally they come to each other like K and log, right? So they interact. There is special, let's say, hole in the molecule, which is fitting by size to the interacting, let's say, other molecule, right? So you have protein, big molecule, and small molecule interacting with it. So, and the shape of this hole and the binding some ligands which are there, like different charged parts of the molecule or neutral parts or hydrophobic, hydrophilic start, they have special type of interaction, yeah? And when you know how this interacts, you can modify this key such a way that even fits even better. So it means you can increase efficiency from one hand. And then until you know the target, you don't know how to modify the key, right? So that's why you need to know the target in order to make better key.
Yeah.
That's it. So you could hypothetically just go start randomly changing the chemical and see how it affects it. But if you know the exact target, you know what you need to build toward.
Yes, that's one point, and I can add another point. Actually, a little bit coming back to your questions or previous question, Benji, about the compounds. Because the funny thing is that in the library we have, there are actually 8 compounds which have the same, let's say, target, or it was so just so described. They 8 of them have the same target, but only one of them demonstrate the effect we show. All others are not, which
Very
means
interesting.
that most likely this is some of additional target.
Yeah.
Yes, most
Very
likely it is, and most likely it is not quite specific for that. If
Yeah,
it is
may I ask-
not specifically decided, yeah, sure.
May I ask a follow-up question to that, which is, it seems like the output that you're measuring is the formation of aggregates, which literature has shown has an association with some of these age-related diseases. But at the beginning of the stream, you guys were talking about how there are these two variables that contribute to aggregates, steady-state level of aggregates in the cell. One is the translational fidelity, and the other is this proteolytic machinery. So
Yes.
with these two compounds that they don't interact directly with the translational machinery. Is there a chance they actually do interact with the proteolytic machinery? And does that matter?
We checked it. No. So we did not do the whole the long study about it, the complicated one, but we briefly checked whether those compounds somehow interfere with the proteostatic machinery. No, at least we could not detect it. And yeah. So what is important actually, so we first measure the translation fidelity, as you properly pointed out, and then we expect that increased translational fidelity improves the folding of the proteins and their functional activity. But the point is that all your proteostatic machinery also consists of proteins. So, and that's actually what we also see in our assays. If we apply the compounds, we see slightly increased activity of the all proteins we check for the effect. So it means that probably it improves the whole cellular proteome in this respect. And you also increases slightly, but still detectably, probably effect of your proteostatic machinery as well.
Yeah, if we come to the numbers, like just to imagine, the normal error rate in human body, when we have, it's all the time our body produce protein, and it makes frequency of this mistake is 10 in order of magnitude -3, -4. So it's let's say every 5,000 amino acid is wrong when it's incorporated in the cell. If we take like a normal average size of protein in our body is around roughly, let's say 1,000 amino acids, for example, if we imagine like this. So it means every fifth protein in our body can be containing error, mistake. So then probability, presence of these mistakes increases chance for this protein to get aggregated, or even maybe it's not possible to fold it at all normal way. So then when we reduce the number of these mistakes, so it means like not every fifth protein, but let's say 1 out of 10 only will have this if we increase fidelity twice. It's will make like a very big impact, I think, for all functionalities of all proteins. That's just to make it more visible for you.
Amazing. Do we still have time, Benji, or should we go ahead?
Yeah, I think there's maybe one more important question that came up continuously through senior review and with the community of VitaDAO is, it seems like the disease you guys are interested in going after is Alzheimer's disease or dementia more broadly. And there's a ton of literature showing that aggregates are potentially causal. And we understand that your work is trying to reduce the number of aggregates by increasing the translational fidelity. One of the things that came up that people were skeptical of in the governance process was there's a lot of evidence that it's post-translational modifications that actually cause these aggregates. And it's those post-translational modifications which cause the aggregates that then potentially cause Alzheimer's. What evidence do you all know of from the literature that shows that it's directly the translational error rate and the translational fidelity that is causal of these aggregates that end up becoming pathogenic?
So probably I can answer. Okay. The point is that when you're talking about these post-translational modifications, when we apply it to the Alzheimer, it's probably tau protein which gets phosphorylated, or it's beta amyloid which is non-properly processed during the Alzheimer. The point is that what I mentioned in the very beginning, that those wrongly over-modified proteins, post-translational or over-modified, those proteins works only at the nucleus for the formation of aggregates. If you checked what those aggregates consist of, that is far from majority. There is so either tau protein or beta amyloid. Mostly it consists of everything possible. Especially often you can meet the so-named heat shock proteins, which are chaperones, which help proteins to properly fold, then components of proteostatic machinery, so it's ubiquitin ligases and so on and so on. So the problem is actually following. If you have a mistranslated and misfolded protein, what happens next? It comes chaperone and tries to help this protein to be properly folded. But if chaperone cannot do it, so if it can do it, then it loses affinity to this protein and goes away. If it is not possible because protein is wrongly synthesized, so it's just not possible to properly fold this protein, then chaperone stays attached to it. And actually, it is already ready to be aggregated. There are already two proteins which are together. And that's why often you meet those chaperones and other components inside these aggregations. So aggregates. What we suggest to decrease level of these mistranslated and misfolded proteins. So in this way, we actually relieve the proteostatic machinery to fulfill more effectively their functions. Because at the end, you have your, so how to say, wrongly modified proteins whole your life. But the problem starts only when you're old. It means that something should accumulate. Something should get more and more wrong, so that at the end you have real troubles with health. And we know what is exactly is it. So we think that we know what is it. That's, let's say, short answer.
Amazing. No, that makes a ton of sense. I think the analogy I'm thinking of right now is like crime and the police. So as you age, there's more crime, and therefore the police have to go and deal with one crime, but then that allows more crime to happen in the streets, and then before you know it, there's chaos.
Yes.
So I think exactly that's the analogy that I'm drawing. So anyway, take it away, Michaela.
Yeah, I think that those are all the questions that I had. Thank you so much, Dimitri and Rashid, for your time.
Definitely. So before everyone goes, I just wanted to remind everyone that anyone can support the researchers' work via Catalyst. So to do that, you would just have to go to catalyst.molecule.xyz. If the project successfully hits its funding goal, then we'll, in partnership with VitaDAO, we'll go to Dimitri and Rashid's respective universities and negotiate an IP agreement, and then tokenize that IP and distribute IP tokens. So anyway, all of these things have to kind of happen in sequence, but yeah. Anyway, just wanted to make sure that people know that they can support the researchers and their incredible work that they did an awesome job describing today. So thank you everyone so much for their attention. And yeah, I think we can close it off there. Enjoy the rest of your nights, guys. Thank you so much. And thanks to everyone watching.
Thank you. Thank you.
Thank you very much.
Thank you, guys.
Bye-bye now.
Pleasure.
Bye-bye, everybody.
Bye-bye.